Section 3B.3.2 Neuroprosthetics continued. By similarly eavesdropping on the brain, could we enable someone, perhaps a paralyzed person, to move a robotic limb or command a cursor to write an email or surf the net? To find out, Brown University brain researchers implanted 100 tiny recording electrodes in the motor cortexes of 3 monkeys. Nicolelis and Chapin, 2002, Soraya et al., 2002. As the monkeys used a joystick to move a cursor to follow a moving red target to gain rewards, the researchers matched the brain signals with the arm movements. Then they programmed a computer to monitor the signals and operate the joystick without the monkey's help. When a monkey merely thought about a move, the mind-reading computer moved the cursor with nearly the same proficiency as had the reward-seeking monkey. In a follow-up experiment, 2 monkeys were trained to control a robot arm that could reach for and grab food. Valiste et al., 2008. Research has also recorded messages not from the motor neurons that directly control a monkey's arm, but from a brain area involved in planning and intention. Mussalman et al., 2004. While the monkeys awaited a cue that told them to reach toward a spot to get a juice reward that had flashed on a screen in one of up to 8 locations, a computer program recorded activity in this planning intention brain area. By matching this neural brain activity to the monkey's subsequent pointing, the mind-reading researchers could now program a cursor to move into response to the monkey's thinking. Monkey think, computer do. If this technique works with motor brain areas, why not use it to capture the words a person can think but cannot say, for example, after a stroke? Neuroscientist Richard Anderson, 2004-2005, speculates that researchers could implant electrodes in speech areas, ask a patient to think of different words, and observe how the cells fire in different ways. So you build up your database, and then when the patient thinks of the word, you compare the signals with your database, and you can predict the words they're thinking. Then you take this output and connect it to a speech synthesizer. This would be identical to what we're doing for motor control. In 2004, the U.S. Food and Drug Administration approved the first clinical trial of neural prosthetics with paralyzed humans. Polak, 2004-2006. The first patient, a paralyzed 25-year-old man, was able to mentally control a television, draw shapes on a computer, and play video games, all thanks to an aspirin-sized chip with 100 microelectrodes recording activity in his motor cortex. Hochberg et al., 2006. Sensory functions. If the motor cortex sends messages out to the body, where does the cortex receive the incoming messages? Penfield also identified the cortical area that specializes in receiving information from the skin senses and from the movement of body parts. This area at the front of the parietal lobes, parallel to and just behind the motor cortex, we now call the sensory cortex, figure 3.24. Stimulate a point on the top of this band of tissue, and a person may report being touched on the shoulder. Stimulate some point on the side, and the person may feel something on the face. The more sensitive the body region, the larger the sensory cortex area devoted to it, figure 3.24. Your supersensitive lips project to a larger brain area than do your toes, which is one reason we kiss with our lips rather than touch toes. Rats have a large area of the brain devoted to their whisker sensation, and owls to their hearing sensations. Scientists have identified additional areas where the cortex receives input from senses other than touch. At this moment, you are receiving visual information in the visual cortex in your occipital lobes at the very back of your brain, figure 3.25 and figure 3.26. A bad enough bash there would make you blind. Stimulated there, you might see flashes of light or dashes of color. In a sense, we do have eyes in the back of our head. From your occipital lobes, visual information goes to other areas that specialize in tasks such as identifying words, detecting emotions, and recognizing faces. Any sound you now hear is processed by your auditory cortex in your temporal lobes, Figure 3.26. If you think of your clenched fist as your brain and hold it in front of you, your thumb would roughly correspond to one of your temporal lobes. Most of this auditory information travels a circuitous route from one ear to the auditory receiving area above your opposite ear. If stimulated there, you might hear a sound. MRI scans of people with schizophrenia reveal active auditory areas in the temporal lobes during auditory hallucinations. Lennox et al., 1999. Even the phantom ringing sound experienced by people with hearing loss is, if heard in one ear, associated with activity in the temporal lobe on the brain's opposite side. Mulnikel, 1998. Association areas. So far, we have pointed out small areas of the cortex that either receive sensory input or direct muscular output. In humans, that leaves a full 3/4 of the thin, wrinkled layer, the cerebral cortex, uncommitted to sensory or muscular activity. What then goes on in this vast region of the brain? Neurons in these association areas, the peach-colored areas in figure 3.27, integrate information. They link sensory inputs with stored memories, a very important part of thinking. Electrically probing the association areas doesn't trigger any observable response. So unlike the sensory and motor areas, association areas' functions cannot be neatly mapped. Their silence has led to what Donald McBurney, 1996, page 44, calls one of the hardiest weeds in the garden of psychology, the claim that we ordinarily use only 10% of our brains. If true, wouldn't this imply a 90% chance that a bullet to your brain would land in an unused area? Surgically lesioned animals and brain-damaged humans bear witness that association areas are not dormant. Rather, these areas interpret, integrate, and act on information processed by the sensory areas. Association areas are found in all 4 lobes. In the frontal lobes, they enable judgment, planning, and processing of new memories. People with damaged frontal lobes may have intact memories, high scores on intelligence tests, and great cake baking skills. Yet they would not be able to plan ahead to begin baking a cake for a birthday party. Huey et al., 2006. Frontal lobe damage also can alter personality, removing a person's inhibitions. Consider the classic case of railroad worker Phineas Gage. One afternoon in 1848, Gage, then 25 years old, was packing gunpowder into a rock with a tamping iron. A spark ignited the gunpowder, shooting the rod up through his left cheek and out the top of his skull, leaving his frontal lobes massively damaged. Figure 3.28. To everyone's amazement, he was immediately able to sit up and speak, and after the wound healed, he returned to work. But the affable, soft-spoken Phineas Gage was now irritable, profane, and dishonest. Although his mental abilities and memories were intact, his personality was not. This person, said his friends, was no longer Gage. He eventually lost his job and ended up earning his living as a fairground exhibit. With his frontal lobes ruptured, Gage's moral compass had disconnected from his behavior. Similar impairments to moral judgment have appeared in more recent studies of people with damaged frontal lobes. Not only may they become less inhibited without the frontal lobe brakes on their impulses, but their moral judgments seem unrestrained by normal emotions. Would you advocate pushing someone in front of a runaway boxcar to save 5 others? Most people do not, but those with damage to a brain area behind the eyes often do. Koenigs et al., 2007. Association areas also perform other mental functions. In the parietal lobes, parts of which were large and unusually shaped in Einstein's normal weight brain, they enable mathematical and spatial reasoning. Whittlestein et al., 1999. An area on the underside of the right temporal lobe enables us to recognize faces. If a stroke or head injury destroyed this area of your brain, you would still be able to describe facial features and to recognize someone's gender and approximate age, yet be strangely unable to identify the person as, say, your grandmother. Nevertheless, we should be wary of using pictures of brain hotspots to create a new phrenology that locates complex functions in precise brain areas. Udall, 2001. Complex mental functions don't reside in any one place. There is no one spot in a rat's small association cortex that, when damaged, will obliterate its ability to learn or remember a maze. Memory, language, and attention result from the synchronized activity among distinct brain areas. Knight, 2007.